All right, everyone, welcome back. We're now at our third lecture for chapter 3, where we're going to now dive into the interior of the cell and look at some of the goings on there and learn how the different structures within a cell actually work. And as always, we're going to begin with our not really attendance questions. So see if you can answer these. What is the primary component of the plasma membrane? And what are the small structures that are found within a cell that carry out specific duties called? Now go ahead and pause the video, try to answer these questions. And hopefully you've done that. So let's take a look to see what we have here. What is the primary component of the plasma membrane? Well, we've actually asked this as a previous not really attendance question. Hopefully it's easier to answer now. Remember, the primary component of the plasma membrane are phospholipids. We have a phospholipid bilayer, and there are other types of lipids and carbohydrates and proteins, but by far the most abundant component is phospholipids. Next, what are the small structures that are found within a cell that carry out specific duties called? They are called organelles. And that is going to be the topic of our lecture today. We're going to take a deeper look at those organelles and see exactly what they do. And we're going to begin with the nucleus. Now, the nucleus really does fit the definition of an organelle, but often it is considered separately from the organelles just because of the way that it behaves and how important it is to the cell. Sometimes, like we saw in the previous lecture, sometimes it's considered a part of the cytoplasm. Sometimes it's considered its own thing, but we're not concerned with how we classify it. We're concerned with what's it look like, what does it do, what is it made up of, what's its structure. So let's take a look at the nucleus. And if you took a biology class in middle school or high school, you might have heard the nucleus referred to as the brain of the cell, because in a way it will dictate what the cell does. It controls the cell. So sometimes you might also hear it called the control center of the cell. But if we want to really say, what does the nucleus do? Well, it houses the DNA. And the DNA is likely the most important thing found within a cell. We will take a much deeper look at DNA on the next slide, and then deeper still when we get to the fourth lecture of chapter 3. So the structure of the nucleus is it is bounded by or wrapped in a membrane called the nuclear envelope. The nuclear envelope. And the nuclear envelope is the membrane of the nucleus. The nuclear envelope is also a phospholipid bilayer. Very, very similar to the phospholipid bilayer of the cell. They are almost identical. There are some extra very important proteins built into the nuclear envelope, but aside from that, it's practically identical to the cell membrane or the plasma membrane. But another really big difference is that it's not just a phospholipid bilayer. That phospholipid bilayer folds back on itself. So it is two phospholipid bilayers. It is basically a 4-layer membrane. And if you look closely at the image here, that black area is the space in between the outer and inner layer of the nuclear envelope. And each layer is a phospholipid bilayer. Now, things need to be able to get in and out of the nucleus, but the nucleus is very, very selective with what can get in or out. Since the DNA is found within the nucleus, and the DNA is so important, the nucleus is very protective of that DNA. It needs to really, really regulate what can get in or what can get out. And scattered throughout the nuclear envelope are these special channels called nuclear pores. And the nuclear pores regulate what can pass through the nuclear envelope. Very, very few things can pass in or out through those pores. Also found within the nucleus is a small structure called a nucleolus. Now, some cells have many nucleoli. Some cells just have one. But the nucleolus, or nucleoli if there's more than one within the nucleus, it is made of protein and RNA. And its function is to make little bits and pieces of ribosomes. And ribosomes we will look at in a future slide in this same lecture. But for right now, we will just say the nucleolus makes the bits and pieces of ribosomes. Ribosomal subunits, we will see it called sometimes. So the nucleus has a nuclear envelope that contains nuclear pores. Inside, we find the nucleolus and DNA. Now, the nucleus, in almost every cell of our body, there is one nucleus. Very, very few cells will have more than one. When we get to the muscular system, we will see that skeletal muscle cells are multinucleated. They will have more than one nucleus. But as a general rule, our cells have a single nucleus. They can have lots of nucleoli inside the nucleus, but generally only one nucleus. Now, like I said, inside the nucleus, one of the things that we find is DNA. And we looked at the structure of DNA and the basic function of DNA at the end of chapter 2. Now, if we were to look at most cells in our body, that DNA is in this hazy state called chromatin. If you looked at a cell, you would not really be able to see strands of DNA. I'm going to back up here, and you can see just kind of this faint haze inside the nucleus. That's chromatin. Most cells in our body are not dividing, and so the DNA is in the state called chromatin. Chromatin is composed of DNA and certain proteins. So let's look at the structure of DNA and how it exists within our cells. First, DNA, here's that double helix that we learned about in chapter 2. Looks like a ladder that somebody twisted. So we have our DNA. But it is very, very important. It controls the recipe for all of the proteins in our body. We'll see what that means in our next lecture. We need to keep DNA very safe. Things like to bind to DNA, but we don't generally want that to happen. So we have some protective mechanisms. Well, first, it's housed inside the nucleus and not much can get to it. So that's one way of protecting the DNA. But next, we have these proteins called histones, or histone proteins. And DNA wraps itself around those histone proteins, like we see here. And if DNA is wrapped around those histone proteins, it joins to those histone proteins, and that kind of makes it harder for other things to bind to the DNA. We want to wrap the DNA around these histone proteins so that the DNA is not going to interact with other things. All right? And that's good. It's now protected inside the nucleus. It's wrapped around histone proteins. But DNA is so important that we need to protect it even more. There's still some exposed areas on the DNA here. So we take the DNA and we start to tightly coil it around itself. This blocks off even more areas of the DNA, making it even harder for things to bind to it. The point of all of this is to make it very, very difficult for things to bind to the DNA. We want to wrap it up, protect it as much as possible. That is chromatin. Now, like we said, most cells in our body are not dividing, and most of the DNA in our body is in that state. But when a cell goes to divide, when it's ready to go into mitosis, like we saw in the lab, that DNA needs to be easy to move around. We need it to be able to be manipulated a little bit. So what we're going to do is we're going to condense it. We're going to take this tightly wrapped coil of DNA and start to twist it and twist it and twist it back on itself until it condenses even further into these structures called chromosomes. Chromosomes are what you see when you look at some cells under a microscope and you see these little worm-like structures. Chromosomes are very, very condensed DNA. It is chromatin that has further wrapped around itself to be as condensed as possible. All right, so now we're going to move away from the nucleus and we're going to look at the other contents of the cytoplasm. Remember, the cytoplasm had two parts. It had the liquid part called cytosol and the little structures called organelles. Now the cytosol, it is mostly water, but it is this kind of jelly-like water because it's got a lot of stuff in it. It's got things called inclusions. And inclusions is kind of everything that doesn't really fit the definition of organelle. One, they're really, really small. Some of them are just like nutrients dissolved in the cytoplasm. But it could be things like fat droplets, stored glucose structures called glycogen. Glycogen is just a type of starch almost that is a lot of glucose stuck together one after another after another. But we're really concerned with the organelles. So organelles, like we said, are kind of like the organs of our body, but in a cell. And we're going to look at each of the organelles and see what their function is. Starting with something called the endoplasmic reticulum, or the ER. The endoplasmic reticulum, or ER. And there's two types of endoplasmic reticulum. There's something called the smooth endoplasmic reticulum, SER, or smooth ER, and something called the rough endoplasmic reticulum, rough ER, or RER. They look very, very similar, except the rough endoplasmic reticulum has ribosomes stuck to it. The smooth endoplasmic reticulum does not. So let's look first at the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, or smooth ER, has a few functions. One of them is this is where various lipid-based components of our body are synthesized. The smooth ER makes certain lipid molecules. It synthesizes things like certain fats. Next, the smooth ER will metabolize toxins. It takes things like alcohol and renders them into a less harmful form by metabolism. We find lots of smooth ER, for example, in liver cells. And lastly, the smooth endoplasmic reticulum stores calcium. I think in the previous lecture, maybe the one before that, we said that calcium is really high concentration outside the cell and practically non-existent inside the cell. Calcium likes to attach to things. And when it attaches to things, it causes certain activities that we don't necessarily want. So we don't want calcium to be inside of our cells. It will attach to things and cause things that we don't want to happen. So if we have calcium inside our cells, it's going to be stored in the smooth endoplasmic reticulum. That is going to be very, very important later in the semester when we get to nerves and muscles. So remember that. Calcium inside the cell likes to attach to things. Normally we don't want that to happen. So if it is inside the cell, it is stored inside the smooth endoplasmic reticulum. All right, so let's look at the rough endoplasmic reticulum, the rough ER. It looks very, very similar, except it's got ribosomes stuck to the outside of it. Well, before we talk about the rough ER, let's talk about ribosomes. We mentioned them when we were talking about the nucleolus. What are ribosomes? Ribosomes are these very, very tiny structures. Some books will call them organelles, other books will call them inclusions. They are really tiny. They're made of two pieces that stick together. And their job, regardless if they are stuck to the endoplasmic reticulum or floating around free in the cytoplasm, because we will find them both ways, their job is to read RNA, ribonucleic acid, and based on what it reads, it makes proteins. So the function of a ribosome is to make protein. Doesn't matter if it's stuck to the rough ER or if it's floating around loose in the cell. Ribosomes make protein by reading RNA. Well, if we look at the rough endoplasmic reticulum, since ribosomes are stuck to it, he will also make protein. The way that that happens, it's actually the ribosomes that are making protein, but that protein is then fed into the interior of the rough endoplasmic reticulum, so that when the protein is finished being made by the ribosome, it is trapped inside the rough endoplasmic reticulum. So let's hold that thought, and we will come back to the rough endoplasmic reticulum in just a moment. Right now, we are going to follow that protein that is now trapped inside the rough ER. When a protein is made and fed into the rough ER, a piece of the rough ER is going to break off. It's going to form a vesicle. We talked about vesicles previously. A vesicle is just a small, basically, package. It's kind of like a bubble or a balloon that's made of pieces of rough ER. It's the rough ER and the smooth ER, by the way, are both made of a phospholipid bilayer. Very, very similar, almost identical to the plasma membrane. Hold that thought also. We will come back to it. So we've got that protein that the ribosomes on the rough ER made. It was pushed into the rough ER. A piece of the rough ER containing that protein has now broken off and now is a vesicle with that protein trapped inside. That protein trapped inside of a vesicle, we see it right here, is going to travel away from the rough ER to the next organelle called the Golgi body or the Golgi apparatus, or sometimes just the Golgi. Golgi body, Golgi apparatus, Golgi. This vesicle coming from the rough endoplasmic reticulum is going to bump up and merge with one side of the Golgi. And the protein that's trapped inside is now trapped inside of the Golgi. Well, that protein that was made at the rough ER was not in a final working form. It was almost there, but not quite. When it gets to the Golgi job is to modify proteins that were made on the rough endoplasmic reticulum. He will add some things to it. He will take some things away from it. But he's going to modify those proteins that were made on the rough ER. And a piece of that bubble is going to move forward and move forward and move forward until eventually it gets to the opposite side of the Golgi, and a piece of Golgi is going to break off, and it has the final protein inside of it. So now we have another vesicle. This vesicle now has a piece of finalized protein inside of it. Now, we just said the job of the Golgi was to modify proteins from the rough ER. It produces vesicles with that protein inside of it. So what's the point of all of that? Well, let's take a look now at those vesicles. Vesicle is kind of a generic term. It's just there inside the cell. It's a little bubble. It's a little package that contains something. But there are different types of vesicles. The ones we've been discussing so far are called transport vesicles. It came from the rough ER to the Golgi, or it comes from the Golgi off to some other point in the cell. So transport vesicles are just kind of generic vesicles that carry something around the cell. Previously, when we talked about phagocytosis, we said that when the cell phagocytizes or eats something, it produces a vesicle. That vesicle was called a phagosome. It's a vesicle produced by phagocytosis. Maybe it's got a bacteria trapped in it. Maybe it's got some other dead cell trapped in it. Whatever that cell ate, whatever it phagocytized, is trapped inside a vesicle called a phagosome. Another type of vesicle, one that was made using this method from the rough ER to the Golgi, and now as a finalized protein in a secretory vesicle, that can be something called a lysosome. I like to say lysosomes are kind of like the stomach of the cell. Lysosomes, the proteins that they have inside are digestive enzymes. Lysosomes are vesicles that contain digestive enzymes. When the cell needs to digest something, a lysosome is what's going to do that. So let's backtrack. Let's see where lysosomes come from. We have the rough endoplasmic reticulum. A Golgi is going to be involved. And we start with ribosomes on the rough endoplasmic reticulum. These ribosomes make something that's very, very similar to digestive enzyme proteins. A piece of the rough endoplasmic reticulum breaks off. It travels to the Golgi. The Golgi takes that protein, that pre-digestive enzyme, modifies it. Once it's finalized into digestive enzymes, it is packaged into another vesicle, and this vesicle we call a lysosome. Well, let's take another look at something that a lysosome can do. Let's say the cell has phagocytized a bacteria. That bacteria is trapped inside a phagosome. We need to get rid of it. Well, a lysosome contains digestive enzymes. It will travel towards the phagosome and they will merge with each other. They will become one big vesicle. That big vesicle is called a phagolysosome. Phagolysosome. What's inside the phagolysosome? Well, the bacteria or whatever that cell just ate, and digestive enzymes. It's going to dissolve that bacteria or whatever the cell ate. Some of that stuff is going to be useful for the cell. Proteins, lipids, carbohydrates, things that the cell can recycle and use for other purposes. Some of that bacteria or whatever is going to be useless. It's going to be indigestible. It's going to be something that can't be recycled, and that's called a residual body. And the residual body trapped inside the phagolysosome is going to travel to the edge of the cell, and we're going to get exocytose. We're going to exocytose the residual body. Basically, the cell ate a bacteria and now it pooped. The residual body is waste. It's dumped outside the cell. It will make its way into your bloodstream and eventually travel to your kidneys to be passed out in the urine. And lastly, very, very similar to a lysosome is another vesicle with enzymes inside of it called a peroxisome. Well, I'm going to tell you something right now that kind of takes students by surprise, and we will look at it even more when we get to Bio 139, and that is oxygen, the same oxygen that we breathe, is deadly. Oxygen is deadly to practically all living things. And all living things develop methods to get rid of oxygen when it shows up in your body. There is only one single reaction in our body that we need oxygen for. And that's going to be the subject of chapter 24 when we get there in a couple more lectures. But other than that one reaction, oxygen is harmful in our body. And the way that our body gets rid of oxygen and other things like it called free radicals is by using peroxisomes. Peroxisomes, vesicles with specialized enzymes inside of them, will find oxygen and free radicals and will take them into themselves, convert it first into hydrogen peroxide and then into water so that it is no longer harmful. Peroxisomes sometimes also will be used to break down certain lipids. In some cases, they will break down certain toxins, kind of like what smooth endoplasmic reticulum does. But primarily, peroxisomes are there to get rid of oxygen and free radicals. So let's take a look at everything that we said on the past couple of slides. We have the smooth ER off doing its thing, detoxifying and storing calcium and stuff, and it was made of a phospholipid bilayer. We have the rough endoplasmic reticulum that makes proteins with the help of ribosomes. It is made of a phospholipid bilayer. A piece of the rough ER breaks off and travels to the Golgi and merges with it. The Golgi modifies that protein. It is made of a phospholipid bilayer. A piece of the Golgi breaks off and either becomes a vesicle of some sort, a lysosome or a peroxisome, or a vesicle that maybe it travels to the edge of the cell to dump something out of the cell. Well, when it does that, that vesicle merges with the plasma membrane. Well, we've got all these pieces of things breaking off, and we have, even in some cases, a piece of the plasma membrane breaks off and moves inward as a phagosome, and a lysosome merges with it. All these pieces breaking off and merging elsewhere seems like parts of the cell would run out or get confused or get diluted or something. But do we know why this is okay, why it doesn't cause a problem? Look at the picture and see if you can figure out why it's not a problem. Well, everything there is made out of phospholipid bilayer. It is completely fine that this piece of rough endoplasmic reticulum became a piece of Golgi and later becomes a piece of plasma membrane, which comes in and becomes a piece of phagosome that becomes a phagolysosome. All of that is interchangeable because it's all made of the same thing. It's all phospholipid bilayer. That was why I asked what the plasma membrane's primary component was during our thinking, or our not really attendance question. It's all okay because it's all moving back and forth and merging because it's all the same stuff. All right, so I said keep in mind what the rough ER definition was because we were going to come back to it. Ribosomes, we said, made protein, regardless of where they were. But there's a significance to the ones that are on the rough ER versus the ones that are floating around in the cytoplasm. Ribosomes that are floating around in the cytoplasm make protein that are going to be used inside the cell. Ribosomes on the rough ER and the rough ER itself make proteins that are destined to either become some sort of vesicle, like a lysosome, a secretory vesicle, a peroxisome, something like that, or that are destined to leave the cell through exocytosis, things like hormones, or that are destined to become proteins that are part of the plasma membrane. Remember all those proteins in the plasma membrane that we looked at? Well, they came from the rough ER. They were packaged, they were sent to the Golgi, they were modified, packaged again, sent to a vesicle. The vesicle becomes a part of the plasma membrane, and now that protein is within the plasma membrane. This whole thing is called the endomembrane network. The endomembrane network. Okay, change gears a little bit, move away from the endomembrane network and vesicles and all of that. We're going to look at the mitochondria. And I like to, in person, when we have this, I'll say, okay, what does the mitochondria do? And everybody shouts out the same thing, and I tell them, well, not really the best definition. Unfortunately, the definition that everyone knows is too basic for our class. What am I talking about? Well, the mitochondria is the powerhouse of the cell, and that's good for elementary school or middle school, maybe even high school, but we want to know really what does the mitochondria do. The mitochondria produces ATP. Remember, ATP is the fuel that our body runs on. It's anytime our body is doing something that requires energy, that energy is in the form of ATP. And it is mitochondria that make that ATP. By the way, even though mitochondria are not part of the endomembrane network, they do have a phospholipid bilayer. This outer mitochondrial membrane is a phospholipid bilayer. And incidentally, the inner mitochondrial membrane is also a phospholipid bilayer. Weirdly, the outer mitochondrial membrane is very, very similar to our cell's phospholipid bilayer. The inner mitochondrial membrane is almost identical to certain types of bacteria. And everything suggests that at one point, long, long ago, there was a cell that tried to phagocytize a bacteria. The bacteria could make its own ATP. the cell could not. When the cell ate the bacteria, instead of digesting it, the bacteria took up residence in the cell. The bacteria, having its own phospholipid bilayer, was trapped inside of a phospholipid bilayer vesicle. It went on producing ATP. Some of that ATP was used by the cell. The cell gets free ATP. The bacteria gets free protection now that it's inside of a cell. So they just kind of took up residence together until eventually it became the mitochondria. When cells divide, the mitochondria go through their own type of division, very, very similar to bacterial division. It's called fission. Mitochondria have their own DNA. Instead of strands like animal DNA, it's in a ring like bacteria. So all evidence points to mitochondria were once free-living bacteria. But that's okay, they don't hurt us, they actually help us a lot. We could never get as big as we are and do all the things that we do without mitochondria. We will in fact be looking at mitochondria and how they produce ATP in our chapter 24 lecture, which this slide kind of gives a little bit of a hint as to what we're going to be studying. We saw this equation back in chapter 2 when we were looking at redox reactions. So this is just kind of a heads up what we're going to be looking at. This is nothing that we need to know for chapter 2 right now, or chapter 3, I'm sorry. We will look at it as soon as we finish chapter 3 and we move into chapter 24. Now, the last thing that we're going to look at is something called the cytoskeleton. And the cytoskeleton is exactly what it sounds like. It's the skeleton of the cell. It's the little things that give the cell structure. And there are 3 different categories for cytoskeleton. We have microfilaments, which are the smallest. We have microtubules, which are the largest. And then the thing in the middle is called an intermediate filament. They're all different sizes, they're made of different things, and they all have different functions. So let's start with the small ones. Microfilaments. Microfilaments are made of a protein called actin. Actin are these little globs right here, but we stick them all together and we get this long string called fibrous actin. And actin producing these microfilaments function, there's a few. Well, one, when we looked at mitosis in lab and we saw the cleavage furrow, well, the cleavage furrow is produced by actin contracting. Muscle contractions use microfilaments. And certain cells that move are doing so because of microfilaments. Next, intermediate filaments. Intermediate filaments kind of form the framework of the cell. They're there to anchor the cell in place. When we talked about desmosomes the other day, intermediate filaments are in play, reaching through desmosomes to grab onto things. It's also tension resistance for the cell. It allows the cell to keep its shape even though when certain pressure is applied to it. And then the biggest one's microtubules. Microtubules do 2 major things. When we were talking about mitosis and moving around the bits of DNA and the cytoplasm, the other organelles, that was microtubules that were doing that. We looked at a structure inside a mitotic cell called the mitotic spindle. Well, the mitotic spindle is made of microtubules. Mitotic spindle is made of microtubules. Also, when things are moving around in the cell, like all those vesicles we just looked at, they're going to travel along microtubules like a railway. Here's a vesicle using something called a motor protein to travel along a microtubule. And there are videos of this taken at the molecular scale that shows these little motor proteins, and it's hilarious. They look like legs, and they take steps, one leg after the other leg after the other leg after the other leg, traveling along the cytoskeleton. Where do those microtubules come from? They come from something called the centrosome. The centrosome is this cloud-like structure, and if we were able to look at this cloud-like structure with more magnification, it would be made of these little things called tubulin, these little small proteins there. They're just all disconnected, but they will grow outward, starting to stick together to each other from the centrosome. All of the microtubules in a cell have one end beginning at the centrosome and then sticking outward from there. In the middle of the centrosome are these little guys called centrioles. There's two of them in the middle of a centrosome. The centrioles are made of microtubules. We can see there's 3 of them stuck together, and they are in a ring with 9 of those units. What do centrioles do? Well, one way that we figure out what does something do is we take it out of a cell and watch the cell to see, now what can the cell not do? When we took centrioles out of a cell, nothing changed. So it appears that centrioles don't really do anything. As a matter of fact, plant cells don't even have centrioles. So we don't really know what centrioles do, if they do anything at all. Except for cells that have a flagella. Cells that have a flagella, or cells that have cilia, these are little projections that stick out of a cell. Well, the base of a cilia or a flagella is a centriole. Not the same centrioles, because there's 2 centrioles here in the centrosome, but we also will find centrioles at the base of a cilia or a flagella. So what are cilia and flagella? Well, cilia are these structures that stick up out of the surface of some cells. And when we see them, there's a lot of them. We see them, for example, inside the trachea, lining the trachea, the windpipe. And they have this motion where they beat rhythmically. And when they do that, stuff moves across the surface of the cell. In our trachea, mucus lines the trachea, and as these cilia move, they move the mucus upwards so that we can cough it out. So cilia, they're very short, there tends to be a lot of them, and they move substances across the surface of the cell. Compared to, I don't actually have an image of a flagellum. A flagellum is very similar. It's a projection that still has the same interior design. However, there's only one of them when it sticks off the surface of a cell, and it's very long. Cilia, they're very short, and there's lots of them. Flagella, there's one of them, and they're very long. And when flagella move, it moves the cell. It's kind of like a rotor that pushes the cell along. There's only one cell in the body that uses a flagella, and that's sperm cells. The tail of a sperm cell is a flagellum. When it moves, it propels the sperm cell. Something that looks like cilia are called microvilli. But instead of little projections sticking up from the cell, These are pieces of the cell membrane itself that fold back and forth on itself. Here, microvilli, their function is to increase the surface area of the cell. It allows it to reabsorb things, or it allows it to secrete things with a very large surface area packed into a very small space. For example, our intestines are lined with microvilli. The last topic that we will talk about is apoptosis. Apoptosis, sometimes it's referred to generically as cell suicide, but a more appropriate name for it is programmed cell death. The reason that it got the term cell suicide is because something happens inside the cell that causes the cell to destroy itself. So why would a cell do that? Why would we have programmed cell death? Well, we look at this gene called PRB. And PRB is the gene that triggers apoptosis. Constantly, a cell is getting ready to go through apoptosis. It builds up and builds up and builds up and builds up, and it's getting ready to do apoptosis. And then there's a checkpoint that says, is everything working the way that it's supposed to? And if it is, then PRB says, okay, I won't do anything just yet. And then the clock resets. And then it starts building up again, building up and building up and building up. And then there's a checkpoint. Is the cell doing everything that it's supposed to do? And if it is, PRB says, okay, I won't do anything just yet. And that keeps happening. But let's say a cell has some sort of defect and it's about to become cancerous. PRB is allowing stuff to build up and build up and build up. And then we have our checkpoint and the cell realizes something's not right here. So everything keeps building up and PRB triggers apoptosis. The cell destroys itself before it can become fully cancerous, because what happens if a cell is cancerous? It divides, it goes through mitosis, and now we have 2 cancerous cells, which divide and go through mitosis, and we have 4 cancerous cells. So a cell going through apoptosis is a good thing when it happens, because it prevents something like cancer from developing. If a cancer develops and grows, that means that there was an error in the PRB gene and it did not get to cause apoptosis, which is what we see here. Here is a cell that's starting to build up these substances. Eventually, the cell starts to get these bubbly areas called blebs, and then it starts to digest itself, and we are left with just this little remnant. So how does it happen? Well, one of the things that happens is we start to make a lot of lysosomes. We keep making more and more and more lysosomes. Eventually, the lysosomes start to burst, and what were lysosomes filled with? Digestive enzymes. The cell is digesting itself from the inside. And in the end, we are left with just a remnant, a dead cell. All right, so that brings us to the end of lecture 3. And that means we have 1 lecture left in chapter 3. And then we move to cover a little bit of chapter 24, where we look in depth at the mitochondria. But next we will be looking at DNA and protein and genes. All right, I hope you enjoyed the lecture. I hope you got something out of it. As you watch it, anything that you're not clear on, send me a question, and I will see you in the next lecture.